Good day, everyone. I am Raymond Tegaraga, your presenter for the research paper entitled Distribution in Abundance of Macrobrachium Populations in Palawan. This paper provides information of the natural distribution and abundance of Macrobrachium in the Philippines and in Palawan. So for the outline of my presentation, number one will be the introduction, followed by the materials and methods, followed by the results and discussion, followed by the conclusion and recommendation, and lastly, the acknowledgement. To give you a short introduction, the Macrobrachium is a genus of freshwater prawns or shrimp characterized by the extreme enlargement of the second pair of periopods, at least in the male. About 100 species more were reported to exist worldwide. This is Macrobrachium is native in the Philippines. While little is known about the population structure of these giant freshwater prawns. While in Palawan, wild catch is available for this resource. Thus, the identification of this resource through the distribution and abundance of macrobrachium populations in Palawan needs to be investigated. This necessitates to the process assessment of various parameters to obtain data for formulating effective management strategy and conservation efforts. Additionally, as considered to be highly priced food commodity, management measures may be developed. For the materials and methods, for the locale of the study, it was conducted in the municipalities in the southern and northern Palawan, Philippines. Prior to the conduct of the study, the researchers conducted coordination through a project presentation with the municipal local government units in the province of Palawan, as well as to the indigenous peoples organization in the province. The study uses descriptive research method. The data were obtained with the aid of structured questionnaire through key informant interviews during the reconnaissance survey conducted in each municipalities to better understand the implication of the project activities in the municipality and vicinities. Structured questionnaires were translated in Filipino or in local language to facilitate better understanding by the informants. Information gathered from the key informants are the distribution, the abundance, the seasonality of the collection of macrobrachium population in the municipality. Photo documentation were also provided during the survey. Data gathered were analyzed and presented into graphs. For the result in discussion, generally the macrobrachium species or the freshwater prawns are present and widely distributed in Palawan's river system, located in different municipalities in the province. Based on the reports, these Macrobrachium species were collected from the rivers of Luzon, Binay, Marikina, San Juan, Pasig River, Manila, Pampanga. Other sources included the streams near the port, Puerto Galera in Mindoro, Taytay in Palawan, Gandara, Samar, Lake Lanao in Mindanao, and Haro in Leyte. Those were in yellow dots. Wild catch of freshwater prawns is also available from the river tributaries in the lakes in the provinces of Ilocos, Cagayan, Pangasinan, Pampanga, Bulacan, Laguna, Palawan, Bicol Region, Leyte, Samar, Cotabato, Maguindanao, Agusan, and some parts of Mindanao. Those are in the red dots. The respondents also noted that the population started to decline during the late 1990s. This can be attributed to the rampant use of various pesticides for agricultural purposes and intensive quarrying activities stretched along the river systems. The abundance of macrobrachium populations is affected by the destruction of their natural habitat, as well as the distribution of this population in Palawan. So the following are the common freshwater prawns collected in the Palawan's river systems that needs to be identified. So we have found 6 commonly caught freshwater prawns. Maybe other are juvenile and some are adults, which are tissues were collected that needs to be confirmed the identity of this species. So the following features were actually collected by the researchers that were noticed to exist in Palawan's river system. So this Macrobrachium SPP species is locally known as ulang, udang, tahi, piyon, orang, and dangawan, to which distributed widely in the river system in Palawan. And belonging or the collectors of these freshwater prawns or Macrobrachium spp species are the indigenous peoples group belonging to the Tagbanwa, Palawan, Batak, Taot Pato, Quirino, and Bolbong. With the increasing threat on the quality of freshwater systems in the localities of Palawan, this impacted to the urgently need of implementation of management measures for the protection of freshwater prawns population in the province. For the conclusion and recommendation, our report confirmed that Macrobrachium populations are widely distributed in the municipalities in the province of Palawan. Researchers also found out that there are limited reports in the catch or for the catch monitoring of Macrobrachium species in the province. Preliminary findings suggest that all municipalities must be visited to continuously map the distribution of the Macrobrachium populations as a promising for potential development of aquaculture industry for the sustainability and food security in the province. Evaluation of other possible human activities to their habitat need to be considered, as well as the management measures should be developed to maintain the impressive abundance of Macrobrachium populations, to preserve the present environment in the river system in Palawan, and as much as fast as earlier, prohibit the use of chemicals to ensure the protection of this species for future generation. So this research paper is part of the research project funded by the DUST-PICARD, the DUST, and implemented by the Western Philippines University, entitled GEMFIL, of the genetic characterization of Macrobrachium populations in the Philippines for broodstock development and seed production. The researchers also would like to thank the local government units in Palawan for their support during the conduct of the study. And that's all. Thank you very much.